I welcome all the participants in our today's seminar. And as you know, this seminar series is part of our DAS event calendar, and it is carried out on every Thursday at 2 p.m. Today, the topic of our seminar is Landscape of Genetic Disorders in Pakistan, Challenges and Opportunities. And it is being presented by Dr. Uzma Abdullah, Assistant Professor, University Institute of Biochemistry and Biotechnology. I hope this scientific talk will provide an opportunity to get in-depth knowledge on the topic. After the presentation, there will be question-answer session, and if you have questions, you can ask the question. Now, over to Dr. Uzma.
Thank you so much, sir. Let me share my screen. Sir, please confirm that my screen is shared.
I think it's fine.
Thank you so much, sir. Good afternoon, ladies and gentlemen. Thank you for joining me here. I am Dr. Uzma Abdullah, and as sir has already very kindly introduced me, that I am serving as Assistant Professor of Biotechnology at University Institute of Biochemistry and Biotechnology, Arid Agriculture University, Rawalpindi. For the next 30 minutes or so, I will discuss the prospects of genetic disorders in Pakistan, its challenges, and among these challenges, the opportunities that I have learned from my own experience in this field. Before going to the scientific part, let me share a brief of my research journey. It started in 2010 when I joined NIPG for my MPhil studies and got the opportunity to work on human molecular genetics lab under the kind supervision of Dr. Shahid Mahmood Baig. During this time, I worked on candidate gene analysis of primary microcephaly. I have also been actively volunteering for prenatal diagnosis of beta thalassemia, and I also gained training and experience in the field of sampling of families with various genetic disorders, especially schizophrenia, intellectual disability, deafness, microcephaly, and many others. Then in 2013, I started my doctoral studies in the same laboratory, and soon won a HEC Indigenous PhD scholarship. My research work of my doctoral thesis was focused on familial cases of intellectual disability, epilepsy, and very interesting and very challenging one, fetal pathology. To conduct the advanced genomic analysis in these families, I applied for and won HEC ERSIP scholarship for the 6 months. And then I also won Lubeck fellowship for 1 year. So during this 1.5 year of my research stay abroad, I worked in University of Copenhagen, Denmark, and also in Kennedy Center, Glostrup, Denmark. There in these two institutes, I learned the techniques of various techniques of genomic and functional analysis. Specifically speaking, these include karyotyping, homozygosity mapping, next generation and Sanger sequencing, molecular cloning, site-directed mutagenesis, and I also worked in human cell culture, immunofluorescence, and immunoblotting. Coming back to Pakistan in 2016, later I joined the same laboratory as senior research associate, and in parallel, I was working on my completing my doctoral thesis, but I also gained the expertise in lab management, developing collaborations with medical community, and field sampling to remote areas, including the far-off areas in southern Punjab. I've been up to Rahim Yar Khan, and also in the northern areas such as, especially in the KP province. Finally, I graduated in 2018, and very soon after, I joined University Institute of Biochemistry and Biotechnology, Arid Agriculture University as assistant professor, and in 2019. And this time has been the time of very active learning and diverse experiences for me. Other than teaching, I have been involved in research supervision, developing collaborations, contributing to scientific write-up, and I'm also serving as reviewer for different scientific journals and external examiner for research students of different universities. So that was a little bit about me. Coming towards today's topic, since I'm expecting that I have non-specialized audience, and I suppose that there must be few people who do not have much strong background in life sciences. So let me give you a simple overview of what genetic disorders are. This is a picture, a simple drawing of a human cell. Uh, each human cell contains hereditary material, that is the DNA. Let me take the pointer. DNA, it lies inside the nucleus. And for humans, the DNA is arranged in the form of 46 chromosomes, as these are detailed here. On each chromosome, there lie various genes, as you can see here. Sometimes it happens that there can be any abnormality in the number or structure of one of these chromosomes. So that can cause two different types of diseases depending upon uh, which chromosome it is or which part of a particular chromosome it is. So some of these chromosomal disorders you must be familiar. These include Down syndrome, Klinefelter syndrome, and Turner syndrome. Other times, there can be loss of function mutations or abnormalities in only one of these genes. And this can interrupt a very crucial biological process and cause diseases such as thalassemia, microcephaly, or deafness, and many others. So such type of diseases are called Mendelian disorders. Um, Still going to another level of complexity, there can be small abnormalities in multiple genes at a time, and these together can predispose a person to developing late onset complex disorders such as diabetes, cardiovascular diseases, obesity, schizophrenia, cancers, and many others. These complex disorders are also called common diseases. On the other hand, the chromosomal and Mendelian disorders, these together are uh, normally very rare in populations. So these are called rare disorders. Coming towards the rare disorders, rare disorders are also called orphan diseases. These are the diseases that affect a small percentage of population. And by definition, it is the one that affects 1 in 10,000 people or even less. It sounds like a small number, but collectively it makes a huge global population of around 300 million people. Among these, the Mendelian disorders are the ones that are of our prime concern. These are the disorders that run in families and they have high recurrence ratio. And these are those that are hugely diverse. So far, at least 7,000 different Mendelian disorders have been identified at clinical level, but only around half of these have their molecular level characterized. That means for rest of them, we do not know the genetic architecture or genetic mutations. And for most of these disorders, there is no treatment. And these can be only prevented, or in certain cases, certain type of limited treatment is possible only if a disease is well characterized at the molecular level. And about these prospects, we will discuss more about it. Before going towards the details of genetic diagnosis, I would like to highlight that for rare disorders, getting even the clinical diagnosis is such a challenge both for the clinicians and for the afflicted families. Parents, when they see a child, they always know that there is something wrong with the child. But when it comes to a child with a rare disease, the search for an answer can turn into an odyssey. Today, it can take an average of 5 to 7 years to get an accurate diagnosis for the patient of a rare disease, even in countries with a sophisticated healthcare system. Too many families around the world bounce between physicians and specialists only to receive multiple misdiagnoses, and the consequences can be devastating. It can cause delay in treatments that could save child's life otherwise. And when there is no treatment or cure, it robs the families of the peace of mind and the options. It creates, there are various factors that contribute towards these complications, such as there is very limited access to, world over, there is limited access to diagnostic tools, and there is especially the shortage of geneticists. Uh, the one most striking feature about rare diseases is that the physicians get to see them so rarely and that they do not have good training in it. And it causes immense burden and psychological distress, both on the families and on the healthcare professionals who are interested with the patient's care and well-being. So the rare disease patients and families may benefit from genetic diagnosis. The genetic diagnosis may not be directly associated with any treatment options, and physicians will continue to treat symptoms, but they will do so in the more informed way. And therefore, the genetic diagnosis could be of benefit beyond the treatment management, and it can also inform the families who just want to know that what is wrong with their kid. And it can also endow the families and patients with the option of making informed reproductive decisions. So coming towards the scenario in our country, Pakistan is a big country of a huge population of over 220 million people. It is sixth most populous country in the world. We have very low literacy rate comparatively, but still, what is most relevant to today's topic is the heterogeneous composition of Pakistani population. There are multiple ethnic groups and language groups such as Punjabi, Pakhtun, Sindhi, Baloch, and then there are language-based groups such as Saraiki, Hindko, Urdu, Potohari, Kashmiri, Brahvi, and many others. And within these language and ethnic groups, there is a very strong tendency to marry within close kins. People favor marriages in close kins due to several beliefs, such as the retention of ancestral property within the family, or there are strong relation bonds. They think it can lead to better compatibility. There would be less divorce ratios and easy marriage contracts. These are few of the many factors. But these are the good things, but there's also a little bit of its side effects. That is, the ratio of consanguineous marriages is so high in Pakistan that it falls amongst the highest. Here you can see the map of Pakistan. This is a consanguinity map of the world, and here you can see the scale that a higher consanguinity is shown with the darker colors. And you can see that Pakistan comes up in the darkest color, and it corresponds to the consanguinity of 50+%. Not only in the world, generally speaking, consanguineous marriages are more common in Muslim world, but even among the Muslim world, Pakistan is exceeding many other Muslim countries in this ratio. Pakistan corresponds to roughly 70% of the consanguineous marriages ratio. So one of the challenges it brings to the nation, to the population, is that it increases the risk of autosomal recessive genetic disorders up to 18 times. And this is the reason that prevalence of hereditary malformations is very high, is proportionately high in inbred or consanguineous populations. So that leads me to the question that how prevalent are genetic disorders in Pakistan? The reality is that we don't exactly know, because there are multiple factors. We do not have any nationwide registry to document such cases, and many of the areas in Pakistan, due to geographical regions or infrastructure, lack of proper infrastructure, these are difficult to access. Also, there is another factor that there is generally, due to low literacy rate and weak healthcare system, there is poor disease awareness in our public. Also, there is poor access to healthcare facilities, and also again comes to the point that rare disorders are generally difficult to diagnose. Even if the parents see or patient, the clinicians see a case, it's not always possible that they can diagnose it and they can categorize it in the category of rare disorders. And this problem is common worldwide. Well, on the top of that, there are many cultural issues of our population. Through my own experience and also through the literature, I can see that majority of people do not actually believe that a disorder, the disorder they're facing or they're seeing, these are of genetic origin. Mostly they attribute it to magic or possession by spirit, especially in the case of epilepsy, you will see it very common. And even if they see that it can be a genetic disorder, they often believe that it's a God's will, and they should not, they do not try to uh, seek the treatment or prevention options. As I have already mentioned, that prevention option is through prenatal diagnosis. But in our society, people have different religious beliefs on the prenatal diagnosis and termination. So that is also a factor. But other than that, there are various stigmas and taboos associated with the genetic disorders. And therefore, even if people have good awareness that someone in their family is suffering from a genetic disorder, they do not like to show it or share it or access the relevant community. And it also complicates uh, the family relations and society overall, that females especially, they suffer more because they're often blamed for having a child with a genetic disease. So these are few of the our cultural populations that are both increasing and hindering the progress in the field of genetic disorders. So the solution for all of this is both uh, investing the efforts on genomic research and diagnosis. The genomic research and diagnosis is very challenging and expensive world over, not only for Pakistan, it's a problem world over, but with the recent down pricing of next generation sequencing, it is becoming more and more cost effective. That is also comparatively still, it's very, very expensive for common people to explore, to afford. And it's also generally time consuming. It takes long time to get the right diagnosis, but still things are getting better uh, in terms of technical facilities in both genomic research and diagnosis. But still, there is no state funded initiative in this field. Whatever work is being conducted on genetic disorders in Pakistan, this is being conducted by a few academic research groups, and they're also mostly relying on international collaborations. And the genetics community of Pakistan today is flourishing, growing, but all of us are very much fortunate and thankful to stand on the shoulder of giants of our field. The pioneer of our fields are the people from Dr. Sheikh Riazuddin from Camp University of Punjab, Dr. Wasim from Qaid-e-Azam University, and Dr. Shahid Beg from NIPG Faisalabad. These are the people who paved the ways and set a research culture and trends that our current generation of geneticists is following, and they have also contributed a lot towards capacity building of our country for expert geneticists who can take on new ventures and explore more of the genetic disorders in Pakistan. Today, many more people are working in this field all over the country, but they are in pockets. They're working in pockets, and still there is no central database where they can share their data, and they can be effective in providing the real-time benefit to these patients. Let me give you an idea of how characterization of a genetic disorder can benefit the families and society. I have chosen the simplest, most example, that is beta thalassemia. Beta thalassemia is a genetic disorder um, of blood. In this disorder, and the beta chain of hemoglobin protein is either reduced, it's not sufficiently produced, or it is completely lacking. So therefore, the patients require regular blood transfusion. This disorder is among the genetic disorders. This is one of the simplest and most well characterized disorder. It's an autosomal recessive disorder. Biallelic mutations in HBB gene, that is one of the smallest gene, cause this disorder, and it's a big problem of Pakistan. In Pakistan alone, annually more than 50,000, more than 5,000 patients of thalassemia are born, and generally speaking, our almost 6% of our population is carrier of this trait. That is, they did not develop the disease, but if they marry, their children have 25% chances of developing major thalassemia major or thalassemia. So, from my experience and the practice that I have learned, I have participated in and I have learned in NIPG, that is, they collaborated with Minar Institute, Multan, and they used to send us the samples of a sick child and along with their parents. We will do the primary screening for the most frequent mutations. These were the 5 mutations that we could detect through simple arms PCR, and these could resolve the mutation in almost 80% cases. For rest of the families, we can go for Sanger sequencing, and we will identify rare or novel mutations. And once the mutation is characterized, that which mutation is running in this particular family, this family can opt for prenatal diagnosis. That is early in pregnancy, during the first, when the first trimester ends, almost at 12 week, they can opt for chorionic villus sampling. For those of you who want to have a better idea or do not know, let me explain that chorion is one of the membrane that bags the developing embryo, and through chorionic villus sampling, the physician inserts a syringe or needle uh, transabdominally, and scrapes off a little parts of the chorion and uh, aspirates it out. And then we can extract DNA from this chorionic villus sample, and then uh, through PCR, we can identify if this baby, if this chronic villus or if this growing fetus has the same mutation that is running in his family. And if it has the mutation, if it is, we see if it is homozygous, and if it is homozygous, then it is going to be a thalassemia patient. So we normally used to return the report within a week or maximum 2 weeks. So they still have time to decide if the fetus is homozygous for the mutation, it's going to be affected. They can opt for the termination of pregnancy. They had this the option of informed decision making then. From my experience, when I was in NIPG, by then we had already characterized around 1,800 couples, and 15 of them opted for prenatal diagnosis later. And it was among them almost 40+% were those that were homozygous affected, while rest of them were either heterozygous like their parents, or they were wild type. So uh, almost 40% of these 15, my maths is bad, I'm sorry, it makes around I think 600 plus, at least 600 plus at least. So these are the births that are affected births that are prevented from only one center or from only one region. So this is the type of impact that we geneticists dream of for all other genetic disorders too, that diseases that well characterized, and we are able to offer them the prenatal diagnosis, and they have the um, they have the option of making an informed decision. Well, the thalassemia is one of the simplest, one of the most straightforward, and one of the most successful example, we would say. But other Mendelian disorders, these are not that simple. Let me give you an example of Joubert syndrome. In this syndrome, patients have gait abnormalities, they have intellectual disability, and they have a problem with coordination of eyes and hands movement. And uh, these are the main features of Joubert syndrome. This is also a Mendelian disorder, just like beta thalassemia. This is also autosomal recessive disorder, just like beta thalassemia. But unlike beta thalassemia, so many more genes are involved that can cause this disorder. At least 34 genes are already known for Joubert syndrome, and mutations in any one of these can lead to Joubert syndrome. This phenomena is called genetic heterogeneity, and the complication does not end here. If we take if we take one of these genes, for example, TMEM67 gene, and see, we observe that different type of mutations in this gene can lead to different type of syndromes. For example, here you see that TM67 already we saw that it causes mutations in it cause Joubert syndrome, but some mutations in it can also cause Meckel syndrome. In Joubert syndrome, the patients live their full life. They are affected, then they're intellectually disabled, but they have the good life expectancy. But in Meckel syndrome, uh, it uh, it's it severely affects the development of fetus. The fetus or the fetus have a hole back at their skull that is called encephalocele. They have multiple uh, fingers that is called polydactyly. They have polycystic kidneys. Their abdomen is often swollen and distended, and they either die before birth, that is intrauterine death, or they die soon after birth. So it is that severe. On the other hand, some mutations in TMEM67 can also called nephrophthisis. In this disorder, only the kidneys are affected and rest of the organs are normal. So in this case, if someone has nephrophthisis, they can go for kidney transplant and they can be okay. And on the other hand, it can also call coloboma. That is only the disorder of eyes and no other organ system is affected. So uh, um, this type of scenario, this phenomena is called phenotypic heterogeneity. That means there's one gene and different type of mutations in the same gene can cause different type of disorders. So we cannot offer prenatal diagnosis for sure until we already know what this mutation is, what precisely what kind of disorder this particular mutation is going to cause. So therefore, it is very, very important that a maximum number of such patients are characterized at the genomic level, and this data is compiled and this data is compared, and the high confidence variants are then uh, 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 brought into practice for providing prenatal diagnosis. And unfortunately, these type of disorders are so common in Pakistan, but they're also least characterized. So, but we still have hope. We have ways. I would like to share a small another success story. This is a huge family of glioblastoma, that is a brain tumor. Mostly it is caused by somatic mutations, but almost around 2% cases are also caused by germline mutations. That is, they run in families, and when their kids reach around the age of 8 or 9, they develop glioblastoma. They're not perfectly healthy before that. At around age 8 or 9, they develop the disease and they die up, I would say, very terrible, very um, miserable death within a year. So this was a family that came into attention. Dr. Shahid Beg and I and many other team members from NIPG who worked on these families. We initially started their diagnosis by sequencing candidate genes that are listed here, but we did not find any mutation. Then we analyzed one of these patients through exome sequencing and identified a mutation in this gene, PMAS-2 gene. And when we had this gene mutation characterized, we offered them prenatal diagnosis. As you can see here, that was also again through ARMS PCR, and we checked 3 fetuses, and all 3 of these fetuses were affected, and they had to terminate the pregnancy. But finally, after at the uh, fourth prenatal diagnosis, it was a heterozygous carrier, and since it was a heterozygous, it was going to be okay. It was not going to develop disease, and it was so difficult, so challenging at that time to get them the fourth prenatal diagnosis. But we made it, and we're so happy to see that it's going to be a healthy child. And when the child was born, uh, the father emailed us and they thanked us and said he's the happiest moment of his life. He also shared the picture of his daughter, and this is was one of the most rewarding experience, and this is what we dream of for our other families too that we work on. So taking from all this experience, I would move to my journey at University Institute of Biochemistry and Biotechnology. Here I joined in November 2019, and I was very fortunate to join a line of very competent people who had already proved their excellence in the fields of human and animal genomics. Starting from our very respected and very dear Professor Dr. Asra Khanum. She has uh, she has performed excellent work on cloning of genes to be used in livestock and human health. And at the same time, she also led the development of diagnostic methods and DNA-based vaccines for tuberculosis. Uh, all of you, uh, ma'am, you are pride of us, for all of us, and a very good example for all of us. We all try to achieve the level of excellence that you have established for us as a goal. Then Professor Dr. Ghazala Kokabraja, she has joined in 1919, and she has done very diverse and huge research on both complex diseases and rare diseases. This included obesity. That was a very large scale study. You must know about, many people must know about it. And then there has been huge studies in metabolic disorders. And one of my very favorite is addiction. She worked on, she is the pioneer of working on genetics of addiction behavior in Pakistani population. She also has been working on rare disorders such as ataxia and other neurodegenerative disorders. And then there comes Dr. Pakiza Arzu Shaikh. She has done exemplary work on many familial cases of skin disorders and epilepsy and schizophrenia. And I have also recently attended a very remarkable seminar by one of her PhD students about schizophrenia. It's, very impressive. Then we have Dr. Sadia in this group, who has been working on genetics and epigenetics of various infectious diseases, and that's also the latest research and a very proud face of UIBB. Then among all these, I joined in 19, and I would like to tell you a little bit about what I have been doing during this time. Starting from recruitment of families, I have almost recruited more than me and my research team, we have recruited more than 80 families of different genetic disorders. Most of these, as you can see here, fall in the category of intellectual disability and neuromuscular disorders. And some of these are deafness, microcephaly, epilepsy. But my major focus, as you can see here, has been on intellectual disability and neurodevelopmental disorders. And then during this time, I have, I could not alienize these families without research funding. So I have been actively involved in applying for grants. So far, I have applied for 19 19 grant applications, 11 of them as principal investigator and 8 of them as co-investigator. 15 of these grants I had submitted to national funding agencies, and 4 of them are to international funding agencies. Also, during my stay, I have reached the publication record, publication count of 22 that are already published, and with the affiliation of UIBBR from families here, I have published 11 papers, 2 as corresponding author, co-corresponding author, and 1 as first author, while 4 of these, 4 of our manuscripts are in pipeline, and the rest of them we are preparing. And cumulatively, I have, these efforts have reached me to the impact factor of 75. Coming towards my work strategy, my work, as for most of other geneticists' work, is divided in 2 parts. There are few tasks that we do in Pakistan. That is the identification of families with genetic disorders. We identify them through clinical collaborations and through our social networks. And then we do the sampling and clinical diagnosis of these, because most of these are from remote areas, and many of them are those that have never seen a doctor before, and they do not know what is the name or diagnosis of the disorder. Then we bring it to our lab, we do our initial candidate gene or variants analysis, and if the mutation is characterized, that that point, we report it back to the family. And if it is excluded for our known mutations or our candidate mutations, we send them in collaboration to whole exome sequencing. Some of these families are resolved at this point, and we again report them back to the family. And if these are not resolved by whole exome sequencing, we go towards whole genome sequencing. And we in through both whole exome sequencing or whole genome sequencing, we uh, sometimes we get a type of genes that have never been implicated in any genetic disorder before. We call them um, novel genes. And to understand how these novel genes cause this particular disorder, we go towards functional analysis and disease modeling. And through this disease modeling in cells or in animal such as zebrafish or mice, we identify the disease biomarkers. And some of the all of these data still compiled. We when we are at the step of disease modeling, we also sometimes need to go back to the family for additional clinical investigations and to better understand the clinical phenotype, and uh, compare uh, the cellular phenotype or animal phenotype that we observe with our with the symptoms in our patients. But some of the families, despite all these efforts, still remain undiagnosed. So this is the work strategy. Working on this, I would like to come towards my area of focus for research. I have been focusing on two type of disorders. One domain is neurodevelopmental disorders, and the other is disorders of reproduction. In the field of neurodevelopmental disorders, I am very lucky to have a very competent team. Many of these members are from UIBB team, as you can see in this slide and in coming slides. I have named the team members from my institute, from my university in green, and international collaborations in blue. So from UIBB, I have the kind guidance and input of Professor Dr. Ghazala Kokobraja, and our MPhil students, Ms. Mariam, Mr. Kamar, Ms. Tanzila, Ms. Faiza, and our research intern, Mr. Ghulam Nasir. They are very, very competent people, very hardworking people. They have recruited very potential families from far off areas such as in southern Punjab and even in Kashmir. And then we have very competent international collaborators such as Professor Peter Nunberg. He's a huge name in the field of genomics, and very, very competent and very kind. Dr. Muhammad Sajid Hussain, who is the group leader in Cologne Center for Genomics, University of Cologne, and Dr. Maria Asif, who is postdoctoral fellow in the same institute. So together we have analyzed around 20 different families of neurodevelopmental disorders so far, and we have successfully characterized 15 of them. We have applied this methods of gene panel sequencing and whole exome sequencing, and so far we have identified 8 novel variants in known genes and 3 novel genes, and also I forgot to mention here 3 known variants in known genes. So this is an example pedigree, and this is a patient of microcephaly. This is how we arrange our data. Just to give you an overview, that these circles represent females and these squares represent males, and the filled symbols, either circular circular square, these filled symbols show the patients. So as you can see in this family, there are 6 patients. Half of them are male or half are female. So this is a summary of findings in neurodevelopmental disorders, but I would also like to share a very highlighted work. In one of these families that we have characterized, we have identified a novel gene. The name of the gene is cectin. The family was segregating severe intellectual disability in 2 patients. They also had speech impairment, and this is sampled from southern Punjab, from Bhakkar, by Miss Mariam. And we analyzed this family, and we found a homozygous missense mutation segregating in this family. It affects, it leads to the substitution of a very highly conserved nucleotide, very highly conserved amino acid. And this is one family. We can, in genetics, we can never be sure about the causation of a disease from one family alone. So we are always looking for checking with others, with other members in the genetics community, if they also have the mutations in the same gene, and with similar phenotype. And one of such platforms is Gene Matcher. We shared our findings on Gene Matcher, and through Gene Matcher, we found that another group in Sydney Children's Hospital, they also have the mutation in this gene, and with very overlapping symptoms. So now we are doing the function analysis on this, and we are trying to figure out the pathway, how this mutation causes this disease. This was such an interesting work, and that I submitted it as an abstract in European Society of Human Genetics. And not only my abstract was accepted, but they also offered me the fellowship that covered my registration fee, part of my travel expenses, and complimentary lunches. So yeah. So very recently, I attended this conference of European Society of Human Genetics that took place in Vienna, the city of Mozart. And here I presented this as a poster presentation. And later, I would also like to share in this conference. It was a very it's dream come true type of experience for me. I got to see uh, many giants in my field that I have known only from their research papers, and I have always appreciated them. One of them was Professor Dr. Alexander Raymond. I went to see him. He was so welcoming, and he was so much interested in Pakistani families, and in collaborating, that we planned a joint research application over there, and that we're going to apply a joint application to Swiss National Science Foundation. And these days we are working on this grant application. So he also offered to host Pakistani students from our institutes for their IRSIP or other fellowships. And they will train them in advanced genomic and functional analysis. So it was a success that I count, and I am very thankful to the management and the administration of our university, who allowed me and facilitated me for this visit to Austria. Coming towards another highlighted finding, we also identified another novel gene, the name for intellectual disability, that is called FEV. The patients are so severely intellectually disabled. And since it is also a novel gene, so we again went to Gene Matcher and identify, and we were very lucky to have found another family from Harvard Medical College, from Professor Tim. We had a Skype meeting to discuss these two families and how we can collaborate and how we can analyze this together. And one of the things I would like to mention, that he was so sentimental. He told us that he has this gene identified back in 2008 in one of his families, and since then he has been actively looking that if there should be some other family with a consistent clinical phenotype and this mutation in this gene, that he can then work together on it. So this also happens to many. I think the genetics community can understand this desperation and this excitement very well. So now we are collaborating with him, and we have advanced very sophisticated and very advanced genomic analysis to dissect the pathomechanism of this gene and these mutations. I submitted these findings as an abstract in American Society of Human Genetics, and that is also accepted. That conference is going to take place in November. I hope I can make it. It's going to take place in October, sorry. So this is American Society of Human Genetics is one of the biggest of the world. Coming towards the second part, and it's only a few slides here. I just noticed it's been 45 minutes. The other domain of my work is the disorders of reproduction. And here I'm working on two phenotypes. One is recurrent pregnancy loss, the other is congenital hypogonadotrophic hypogonadism. The recurrent pregnancy loss is actually the failure of two or more pregnancies. It afflicts 2 to 5% couples globally, and mostly it is due to anatomical and immunological reasons or endocrine abnormalities, but it only explains only 50% of the cases. And among 27% of these, we can identify chromosomal abnormalities, but rest remain undiagnosed. And we have very strong belief and evidence that rest of these are caused by biallelic mutations. And this is what we are going to work on right now. Our strategy is that we will sequence the parents and abortus trios. And we are also expecting many novel genes, since this field is in its infancy. So we will do the mouse modeling for the candidate genes. We also have another domain where we would do the case control association studies of RPL for candidate SNPs in our population. For this work, we have a huge group. It's a multi-center study. We have co-PI Dr. Tahir Naim Khan from NUMS University, Rawalpindi. And we have the support of clinicians from PIMS Islamabad, PIMS Hospital, and First Umar Hospital from Chenabnagar. We have very competent team of UIBB, Professor Dr. Ghazala Kapraja, Dr. Pakiza Azushai, our PhD student, Dheem Sikandar, Mr. Imran. He's also my PhD student, Ms. Jesra. She's MPhil student, and then we have research intern who is very active student who has recently joined us. For international collaborations for genomic analysis, we have collaborated with Professor Dr. Neil Stomer. It's a very huge name in the field of genetics. And for modeling work, we will be taking mentorship and facilitation from Associate Professor Dr. Erica Davis. She's group leader, she's associate professor in Children's Hospital of Chicago, United States. And this group is going to grow more as we recruit more research associates and further PhD students. So for this work, we have funding from university. For part of this molecular investigation, we are funded by our university under Research Endowment Fund. And for rest of it, and especially for the functional part, we have funding from NRPU, from Higher Education Commission, and hopefully we will make the best use of it. Coming towards the last domain, that is congenital hypogonadotrophic hypogonadism. This is the disorder of pubertal development. The patients have abnormal synthesis or secretion of gonadotropin releasing hormone, and therefore they cannot make luteinizing hormone and follicle stimulating hormone. Therefore, they fail to achieve puberty, and this leads to infertility. Some of these patients also have associated abnormality of anosmia, that is the inability to smell. The patients have huge psychological burden and very compromised quality of life, but let me emphasize that it is treatable if diagnosed timely. And this is also a genetic disorder, and through genetic characterization, they can be helped. They can be identified pre-symptomatically before time, and they can benefit from timely treatment. For this work, we have it's also a multi-center study. We have Dr. Amreen Fatma, assistant professor from Aga Khan University, and we have from UIBB, we have MPhil student Mr. Ahmed Faraz. From clinicians, we are working with Dr. Umar Yusuf Raja and Dr. Umar Yusuf Raja and Dr. Sama from Shifa Hospital, both of them very competent endocrinologists. And from international collaboration, we have the collaboration with Professor Dr. Nelly Petlau, who has recently visited our institute too, and Dr. Mohammed Ansar, who is also group leader. Both of them are working in University Hospital Lausanne, Switzerland. Dr. Nelly was very kind to visit us recently, and with huge support from our director, Professor Dr. Mohammad Javed Asad, and our worthy Vice Chancellor, Professor Dr. Mohammad Kamruzama, we organized a very successful hybrid seminar, and mm, there was huge participation from university, and many of you must know about it. And so coming towards the way forward, uh, I plan to set up a human cell culture in our institute. That is the domain that I think we are lacking. Um, and we have to rely on our international collaboration. But now, right now, I think we have enough expertise that we can start this work indigenously. We are also, as a part of our project of recurrent pregnancy loss, we will develop the mouse modeling here. And one of our main mandate is about creating awareness about genetic disorders. But more than that, what I am most thrilled and uh, uh, fascinated is to work on is contributing towards the agriculture side, since it is our university is an agriculture university, and I truly believe that my work and my expertise can be uh, can be of use to many of the other faculties too, such as faculty of sciences, faculty of veterinary and animal sciences, and faculty of agriculture sciences. There I can contribute in the genomic analysis of both animals and plants, livestock, and I'm open to the opportunities, and very much looking forward to have collaboration with you guys. So that is the end for me, but I would highly like to acknowledge the contribution of many people in my journey so far. I am extremely thankful to worthy Vice Chancellor, Professor Dr. Kamrisama, for his very diligent efforts to strengthen the research culture in this university and his exemplary leadership. He has been constantly guiding us and encouraging us towards exploring new horizons and supporting our new ventures through his personal commitment and through engaging this university faculty and leading us together as a team. At the same time, I cannot thank enough the leadership of UIBB Director, Professor Dr. Mohammad Javed Asad. He has been so kind, so diligent. He has always promoted and encouraged in the innovative approach, both in the research and in the teamwork. And I am very impressed by his commitment to strengthening and progress in this institute. I would also like to say special thanks to all faculty members of UIBB, each single one of them, starting from the most senior Professor Dr. Ghazala, Dr. Javed, our very dear Dr. Firoza, very kind Dr. Shiraz, and very competent Dr. Sadia, very dear Dr. Pakizar Zashaik, Dr. And very competent, very dear ones, Dr. Taiba, Dr. Bilal, and Miss Hina, and the one who has recently joined, Miss Saima. I'm indebted to all of you. This was my first job, I would say formal job. You all have been so welcoming and so warm and always so supportive in guiding me and through all my work. I'm also very much thankful to my international collaborators for their generous support and guidance. I'm personally very thankful to all the families who have agreed to participate in our studies, and to the clinicians who have facilitated with their diagnosis and with the recruitment of these families. Also, I am highly thankful to administration of Arid Agriculture University. They have been so supportive in whatever a new venture I have I have tried to take on. And the recent example, my recent experience about organizing this international seminar, I have seen the level of competence and the level of support they can offer. So highly impressed, very thankful to all of them. I'm also highly thankful to Higher Education Commission Pakistan, who has funded my project, and also to Arid Agriculture University, who has given me the other funding for the other project. Well, I thank you all for your attention. If you have any question about genetic disorders or my work, or if you have anything in mind that I think I can benefit with, you're most welcome to visit me or contact me at my email address. Thank you all very much for your attention. If anyone has any question, I would really like to answer. Yes, I think Dr. Farooq, you're saying something, but your mic is mute. Dr. Farooq, may I have the questions? Okay, I think he's not in the session anymore. I see Dr. Saifur Rahman has raised hand. I'm sorry, Dr. Farooq has just informed me
Hello.
that he has.
Hello.
Yes.
Can you
Dr.
hear me? It's Dr.
Yes,
Moin.
yes, I can hear you.
Okay. Thank you, Dr. Uzma. It was really wonderful to listen to your presentation, and I really appreciate and wish you good luck for your future endeavors in research. It's just you can say comment or sort of question from my side. In slide 27 and 31, I have observed that there seems no family history. This in this slide, both male and female kids they are affected, and there is no family history. So I was wondering, is there these pregnancies are late pregnancies? I mean, after 45 years of age, that certain mutations all of a sudden appeared in the germline and the kids were affected, or
Yes.
was it already known this sort of mechanism that it is in the lineage? What's your take on this? And if it is so, do you think that if these is affected by single gene, then why not to go for the allele specific testing of these parents or offspring? Because in livestock population, we generally use the allele specific primers rather to go
Yes.
for the, genomic studies or other expensive, you can say, activities. And allele specific primers, they can exactly tell you where is that or where is that mutation is absent, whether the individual is carrier or affected in both cases. Your take, please.
Well, thank you so much for your very relevant question, very good question. Sir, I would like to highlight that this is a family of autosomal recessive disorder. In autosomal recessive disorders, the parents are asymptomatic. They are only the genetic carrier, but for a disease to develop, they have to have both of their alleles mutated. So here, this is the reason that the parents or their grandparents these generations do not have the disease because these are the carrier. They do not have biallelic mutations. But after this consanguineous union, the mutation is inherited in biallelic state. That is why these are the patients. That is when the disease appears. And coming towards your second question, if I understand it correctly, you have asked that how early these can be detected. Am I right?
No, I was talking about the different techniques which we apply for
Oh,
the
okay.
detection of the genetic disorder, and
Okay.
one of that which we commonly use in livestock is the allele specific primer.
Exactly, sir.
For through PCR, you can do it. Yeah.
Yes, sir. Yes, sir. You're right. We can use and we prefer to use the allele specific primers only when the mutations are characterized. As you can see in the example of beta thalassemia, here the arms, this is allele specific primer. So when a
Yeah.
genetic disease is characterized to this level that all the mutations are known, then we can go for the allele specific primer. But in diseases like intellectual disability or Joubert syndrome, for example, there can be any mutation in any gene. So we cannot rely on allele specific primer. I mean, how many primers we can design or how many PCRs we can set up. Even if we try one for each, these are at least 34 different PCRs. And you know, the genes are huge, so it would not exclude the possibility. If they're negative for all of these, it would still be possible that there is some other mutation in some other, in even in the same genes, and that can cause disease. Therefore, genomic sequencing instead of allele specific PCR is the need of the time. But yes, we dream to reach that level where we know the disease so well that we can use only allele specific primers.
So sequencing would be, if you're going for sequencing, you would be going for the specific gene sequencing, right? If I'm not incorrect.
If when the mutation is characterized to that level, yes, we will sequence only that gene. But if the gene is, if the disease is heterogeneous like this one, it is, we rely on, in such cases, we rely on gene panel sequencing, where we sequence only the genes that are already known to be involved in the disease. For example, for microcephaly, we have 27 genes that are known, that are reported to involve. So instead of sequencing all 20,000 genes, we sequence only these 27 genes, and it is also most of the cases. So it's also cost effective and quicker. But this is where we start.
Yes.
Yes.
Thank you, and good luck for your future work.
Thank you so much. Thank you. I also see that Dr. Asra Khanum has raised hand. Please, ma'am.
I just want to comment and advise
Yes,
to give you. It was
Yes,
very impressive work which you have
Thank you so much.
presented, and very good work which you have done. And I believe that you are following the right path as Dr. Shahid Beg has done or has followed. That is, the path is collaboration with national and international organizations, because that is
Yes,
how, because single person cannot do with his or her own what you say, team.
exactly, ma'am.
He or she need to have wonderful and very competent people in collaboration, international and national both. That provides you a huge opportunity, and I can see within a very short period of time you have been able to show your important what we can say characteristics, to be able to do good work along with other people. So one thing as an advice, to be very focused. You have to be
Yes.
focused. You don't go here and there, and you have to be very focused. And at the end, very good luck. I'm so proud of you. Thank you very much.
Thank you so much, ma'am. This, your words, your encouragements mean huge, a lot to me. This is very energy giving. Thank you so much.
You're welcome.
Uh, do we have any other question or should we wind up? Okay, over to Dr. Farooq. I think your mic is working right now, sir.
Thank you, madam. It was really very nice presentation. I really thank you
Thank you.
for very interesting and informative talk on the subject. And I was doing the comments of the participants. They are all also appreciating the work and the way you have presented your subject. I think it was wonderful and we really appreciate it. So at the end, I would like to thank all the participants for their participation. And I hope such a presentation will come in future as well from our part, rest of the participants and presenters. Thank you very much.
Thank you so much, sir. Thank you, everyone.